It is a very great pleasure and a distinct honor for me to introduce Gerald Fischbach today. Jerry is a legend in the field of neuroscience. I knew about his work when I was a graduate student, and then I met him several times as he led multiple institutions, culminating in his stewardship of the Simons Foundation Autism Research Initiative, or SAFARI, when he joined the Simons Foundation in 2006. He subsequently became the foundation's first chief scientist and fellow, and currently holds the title of Distinguished Scientist and Fellow. I want to just say a few words about Jerry's scientific contributions, and then about his influence on the field of autism, and especially the Simons Center for the Social Brain at MIT. Jerry was an early leader in studying the formation and maintenance of synapses, an interest that has continued throughout his life, as evident in the title of his talk. He developed the use of cell cultures to study the electrophysiology, morphology, and biochemistry of developing nerve, muscle, and interneuronal synapses. Perhaps most significantly, Jerry pioneered the search for motor neuron molecules responsible for regulating the number of acetylcholine receptors on postsynaptic muscle. This culminated with the isolation of the ARIA, A-R-I-A, acetylcholine receptor-inducing activity protein, which is a member of the neuregulin family, as we now know, and is responsible for stimulating the production of acetylcholine receptors in postsynaptic muscle tissue. After starting his faculty career at Harvard, Jerry held a string of increasingly influential leadership positions, including being chair of anatomy and neurobiology at Washington University School of Medicine, chair of neurobiology at Harvard Medical School, director of NINDS in Washington, D.C., dean of Columbia University Medical School, and then director of SAFARI, or the Simons Foundation Autism Research Initiative. I think it is fair to say that SAFARI would not have succeeded without Jerry's extraordinary vision and insight. He started several programs that have shaped the field of autism research. He started the Simons Simplex Collection, taking the view that individuals who are the only individuals in their family diagnosed with autism will provide unique insights into the architecture of autism, especially the contribution of rare de novo genetic variants. He championed the Simons VIP and now the Searchlight cohort to take a genes-first approach to understanding what certain variants, starting with the well-known 16p11.2 copy number deletion, contribute to autism. He encouraged the Simons Foundation to expand beyond autism, and he initiated the Simons collaboration on the global brain to bring together computational neuroscientists and system neuroscientists to study brain function. But it's when we approached Jim Simons and the Simons Foundation for expanded support for autism research at MIT that we realized the full force of Jerry's thinking and vision. In 2009, he suggested that we start a seed grant program and a postdoctoral fellows program to initiate collaborative research on autism broadly across MIT and begin to train the next generation of researchers. A couple of years later, when we inquired about expanding the program into a full-fledged center, Jerry suggested that we study the social brain. He asked me to bring together interested faculty, and some of you are here. I see Nancy and I see Bob Desimone. And he led a discussion on how understanding the social brain is one of the next great frontiers of neuroscience, and in fact will be key to understanding autism. Of course, MIT does not have a medical school, and this resonated with us to study a fundamental function of the brain rather than a disease process per se. Jerry encouraged us to think outside the box in framing our programs, and I'm delighted that in the 12 years of our existence, the Simons Center at MIT has had some impact via our highly collaborative research programs and the postdoctoral fellows we have trained. So, as beneficiaries of Jerry's scientific vision, his extraordinary kindness, and his unwavering support, I'm delighted to welcome him here today. Thank you, Jerry.
Thank you. Thank you, Mriganka, very much. It is such a joy to be back here at my favorite academic center in the world. I'm going to give you a brief overview of my career and then end up talking about the future. So first, I began interest in science and, as Mriganka said, synapses at Cornell Medical School when I took a year leave of absence and worked on use and disuse at the intact neuromuscular junction. Then I moved to the NIH, and then we moved to Harvard Medical School, and then we moved to Washington University, and then I moved back to Harvard Medical School, and then I moved back to the NIH, and then I moved back to New York, but to Columbia, not Cornell. So you could look at this and say, this guy has a movement disorder. How could he be doing science? But the sequence was not brief. There were about 8 to 10 years at each place. And each one was remarkably rewarding. And now, since 2006, I've been at the Simons Foundation, where I will spell out some of the things we're doing. And I want you to all realize that none of this would have been possible without the cooperation and collaboration of Ruth, who's sitting here, who pursued a career in social cognition and neuroethics, obtaining her PhD along the way and starting several programs at each place that I worked. I hope you will tell me if my voice is fading and you want me to speak louder. Just jump up and down and scream, and I will do it. But Ruth was integral every one of these steps. I became interested in the neuromuscular junction for several reasons. This is a photo I love from Mildred Kuhn in the 1860s, and it points out that the nerve terminal on the muscle in most striated muscle is restricted to one area. There are arborization of the terminal. And the one thing I really would like you to note is the accumulation of nuclei. These are muscle nuclei that accumulate at the synapse, and they have important functions there in terms of the trophic interactions. Why start at the neuromuscular junction? It is, in my view, the prototypic synapse. It's accessible. Muscle can be dissected out and maintained in vitro for several hours. And there's a remarkable history of research which paralleled the action potential definition. But in this case, the leading authors were Bernard Katz, Stephen Kuffler, Jose del Castillo, and others. But it provided a great beginning. There are well-defined genetic lesions and precise genotype-phenotype correlations. And more, there are potential therapies, I hope, coming soon for neuromuscular and spinal cord disorders. But I was not satisfied with looking at nerve muscle junctions in vitro, which stayed safe and alive for several hours outside the body. So we turned to tissue culture with the help of Mark Namaroff, a student of Howard Holzer, who was nearby in Washington, and dissociating cells from the pectoral muscle, these mononucleated cells called myoblasts, and watched them over the next few days fuse to form myotubes, some of which would contract and which provided wonderful targets for spinal cord neurons. Many people have been growing muscle cells in culture, but very few had ventured into the idea of forming synapses in vitro. So these are all in chick embryos, dissected the spinal cord from E7-day chicks, strip them of the dorsal root ganglia, then minced up the tissue, and then homogenized it by mechanical dispersion into single cells, and plated those cells in tissue culture dishes. You have to realize this was a real breakthrough. The tissue culture dishes were coated with collagen, and the cells settled very easily on that collagen-coated surface. And within time, the neurons, the motor neurons, reached out with axons and dendrites and contacted the muscle cells. And by placing electrodes in the muscle and nerve, you could evoke an action potential and the synaptic response. I was alone in my lab at the time, a dark, small room, about 10 by 10. And when I first saw the first synaptic events, I was very excited. The synaptic potentials were prolonged. They were all due to acetylcholine, blocked by curare, enhanced by anticholinesterases, and they were slightly longer than usual. The mean channel open time at these early embryonic junctions was long. But there were clear end plate potentials, and during repetitive stimulation, there was quantal fluctuation, and you could calculate quantal contents, which were low, in the order of 5 to 10, not 300. And the frequency of spontaneous potentials was quite low. But what really triggered my career was looking for the acetylcholine receptors that were probably accumulating in the muscle fibers. And we did this in two ways. The first was by ACH ionophoresis. Possible to fill a microelectrode with molar concentrations of ACH. And since ACH is a cation, one could eject the ACH with brief positive pulses, and to hold it back with a steady DC current, but the brief pulses elicited sharp responses. And you can see along here, there's a variation. It's low levels for most of the distribution, but in this case, there was one peak sensitivity, which was 10 times as sensitive as the remainder of the fiber. And we call these hot spots. We developed a well-known venom to further chart the distribution of sensitivity. We used venom from the snake Bungarus multicinctus. Have you heard of this venom? It's a peptide shown here, which is very specific for nicotinic ACH receptors. And it's essentially irreversible. Once bound, it stays bound. And we could chart the distribution of receptors not by autophoresis, but by autoradiography after labeling the Bungarus toxin with iodine. And you can see the result here. There's a huge increase in the number of grains over muscle cells that are treated with extract of chick spinal cord and muscle. And we use this Bungarus toxin binding as an assay in further studies. This is me. I was happy then. Relaxing at my desk at Harvard Medical School. I loved smoking a pipe that quickly got vetoed at home, but things were going very well. And I loved my time there. We developed an assay for purification of whatever it was in the chick extract. We plated the muscle cells not in 35 millimeter plates, but in 6 millimeter wells, 96 wells to a plate. And then we would block all the receptors and wait hours, up to 8 hours, 10 hours, for the receptors to reappear. And we documented that the reappearance was due to newly synthesized receptors inserted into the membrane. And what we found with added extracts, fractions from chick brain, is that the rate of appearance of new receptors was greatly increased. And we used this assay to purify from the crude extract, what is it in the principle that is doing this? This was a long effort. It took more than 10 years working at Harvard, at Washington University, and then back at Harvard. And it involved a very talented group of postdocs. Doug Falls, Gabriel Corfas. The guy who really advanced it was a graduate student at Washington University. And finally, Ken helped with the final purification and cloning of the gene. Al Sandrock, who worked here at Biogen for many years, did a lot of the cell biology, charting the distribution, as did Gabriel Corfas. But the final result was the isolation of this clone, which had 602 amino acids, and some key domains. One, an immunoglobulin-like domain. Two, an epidermal growth factor-like domain. Then a transmembrane domain, and the intracellular compartment. I thought, that the EGF-like domain was the active principle. And in fact, when we cut that out of the molecule, we got this tremendous stimulation all by itself of this small peptide, by the small peptide of acetylcholine receptor synthesis. We reasoned that the IG-like domain served to anchor the molecule in the synaptic cleft, binding to cleft proteins. And when we looked at the biology, made antibodies to the molecule, sure enough, different parts of the molecule all turned up at the neuromuscular junction. This is probably bungarotoxin binding, and this is ARIA binding. We call the protein ARIA for the acetylcholine receptor inducing activity. And we found that the protein was expressed in the spinal cord in motor neurons. And it was expressed as early as embryonic day 4 and right up through birth. So all these articles together make me convinced that ARIA is a key molecule in the formation of the neuromuscular junction. Now there are other molecules in this category. They're called neuregulin, epidermal growth factor, and others. It's terrible when your daughter gets married and someone changes her name. It's worse when you work on a protein for 15 years and someone changes its name. But right now, ARIA is gone as a name, and this protein is known as neuregulin 1. My work was not restricted to ACH receptors. In fact, other people in the lab worked on glutamate, GABA, and epinephrine. This is an example from Kathy Dunlop. Showing that norepinephrine decreased the calcium component of this complex action potential in sensory neurons. But others, David Farb, Dennis Choi, Ann Mudge, and Alkman Chalzanidis, worked on glutamate, modulators, and desensitization. We were, I think, among the very first to show that chlordase epoxide facilitated GABA responses in a very unusual way. And that was very rewarding. When we moved to St. Louis, we lined up along the arch, and this work continued. I'm showing this because these people have all been very successful. Ted Usden is a senior investigator at the NIH. Chuck Zaremski, who worked on glutamate receptors, is the chairman of psychiatry at Washington University. Rich O'Brien, I wish I had a pointer here, is chairman of neurology at Duke University. Lorna Roll is a chief scientist at the NINDS. And Larry Trussell is a very distinguished senior scientist at the Volum Institute in Portland. After all this work, I went back to Harvard. And there was welcomed as part of this tradition, which began with Steve Kuffler, followed by Torsten Wiesel and David Potter, and then me. And I was followed by Carla Schatz and Michael Greenberg. A lot was done in these days. Steve became a good friend, as did Torsten and David. But the department had slowed down, in my view. There was one promotion to tenure in that department in 26 years. But when I became chair, we recruited 9 people in the 10 years I was there. And 7 of them were appointed to tenured positions. We reached out to the hospitals in a way that the previous department had not. I spent my first year in Boston at the Mass General Hospital in Charlestown because my labs were not yet finished. So I got to know many people at the hospital, and we recruited people from the hospital, from Children's, Mike Greenberg, and from MGH, Gary Yellen. And it was a very wonderful opening and embracing areas that were essentially excluded from the department. The department expanded in a wonderful way. We initiated a program called Mind, Brain, and Behavior, which opened up the department to the university. MB&B became the second most popular undergraduate concentration at Harvard University. And the idea was to bring in people from other schools, from public health, from the business school, and others, to be part of the neuroscience community. And I enjoyed that and loved it. It was reaching across the Charles River. And I think that's been maintained ever since. But things come to an end, and after 10 years, I went back to the NIH. Harold Varmus recruited me to run NINDS. And several things I just want to point out before leaving that. First, again, the goal was to bring people together. And one of my ideas was to bring all the different institutes, aging, drug abuse, mental health, and neurology, into one community. And in fact, the NIH leadership agreed, and we created the John Porter Center. I don't know if any of you have been there, but it's a beautiful building at the far end of the campus. And this is John Porter, who was a senator, was a strong advocate for neuroscience research, NIH research in general, at a time when NIH funds were tight. In this picture is also Steve Hyman, who is a colleague and is again a colleague at Harvard at the Broad Institute, and Alan Weissman, head of drug abuse. But we brought institutes together and they've remained collaboration, working in the same building side by side. I want to take a bit of a deviation if there's time. One thing that I became obsessed with at the NIH was Parkinson's disease. There was a huge influx of funds for this. And as you're all familiar, there's a loss of dopaminergic neurons in the substantia nigra. People at Emory University developed a technique for inserting electrodes into the human brain, an area of research I think will grow in the coming years. Intervention in the human brain. And this is a movie made of one patient, Sybil, who I got to know very well. Can you hear that? By the time they met Sybil, she was really disabled, as you can see. Her muscles froze. I presented Sybil's story to Congress several times. This is located in the subthalamic nucleus. So there are a lot of these operations performed today, way more than 500 a year. I think you're ruled out if there's significant dementia, but otherwise, many people are accepted into this surgery. And it's a lesson that I hope is learned. Efforts are being made now to treat malignant depression with deep brain stimulation, and also obsessive compulsive disorder with DBS. And I would not be surprised if other conditions follow suit. But after a time at the NIH, it was time to move back to academia. And Ruth and I moved up to Columbia, where she had a great position, and I was the dean of 4 schools: medicine, public health, dentistry, and another. But the common, and I'm not going to dwell long on Columbia. I got to know an academic medical center from the inside out. I know the interactions, complex, tense interactions between the university and the hospital, that time still known as the New York Presbyterian Hospital. The relationship was fragile and tense, but it worked after a while. But after 6 years, I was ready to step down, and I did, and met Jim Simons, and was recruited to direct their autism research program. This is Jim and Marilyn Simons, both of whom I consider among my best friends and mentors I've had during my whole career. Jim is a wealthy man, having invested wisely, but his life is not without trials and tragedy. 2 sons killed in accidents, a daughter on the autism spectrum. So it's really remarkable that Jim and Marilyn dove in to the Simons Foundation research efforts that we initiated. The effort today is concentrated in math, physics, and biology. My part was in the biology part. Jim and Marilyn exert extraordinary leadership, coming to most of our meetings, contributing and asking questions. They encouraged our growth from 7 people to over 400. They provided funds, and they encouraged our use of RFAs. So my chief efforts were in programs called Safari, the Simons Foundation Autism Research Initiative, which collected 2,000 triplex families, 2 parents and 1 sibling on the autism spectrum, exhaustive phenotyping done at 12 different academic centers around the country, where each group was trained by Catherine Lord, who led this effort. Mriganka mentioned other collaborations in the global brain and plasticity in the aging brain, which I also had a role in initiating. There are several challenges facing us, although I'm optimistic. Low NIH funds, low public trust, low public understanding of the science, low respect for basic science, and of course, the mystery of the human mind. Low NIH funds is getting to be a chronic problem. The low public trust is increasing, unfortunately. The low public understanding is also increasing. And some of that is our own fault. We don't explain things very, very well. And the low respect for basic science is not, to me, understandable. And you all are aware of how difficult this is to communicate about the human mind. So in the end, and this is a picture of me standing, Nancy knows this region, in Woods Hole on our property, looking for one of the most gorgeous sunsets possible with an inset of a dendritic arbor. This has remained my focus, and I hope it will for a few more years. And I hope we can communicate a lot more about these efforts. Thanks. So I could try and answer a question, if you have any.
I have one.
Mriganka.
How might we increase public trust in science? And what role might either academics or foundations have in a concrete way?
Mriganka, the only way I can think of is by communicating better. When you're asked to speak at a public function, you think about it very hard and understand that your audience is not as sophisticated as you think. And take your time to explain each concept in some detail. It's amazing how people respond to that. And it's also amazing how little this is done. I know a few people who do it very well, but it means just being vigilant and not becoming combative, but welcoming the questions and trying to understand where they're coming from and just hoping for the best. You know, when you see things which I'm sure everyone in this room is repelled as much as me by the Speaker of the House fiasco and many other possibilities, by fringe groups and overly ambitious people. You have to combat this wherever you can. Nancy combats it on a local level, and I think I admire that very much. Any others? Okay. Thank you.